I'll get rolling. Thank you very much. It's it's nice to be here again for probably about the 8th time at the Stem Cell Conference in different capacities. I'm representing Orbsen Therapeutics, which is out of Galway, Ireland. We're also a stromal cell therapy company. We don't like to call ourselves an MSC company. We think we're a bit distinguished from the MSC field as it's come today. We think we're developing what we call the sort of next generation technology stem cell. So is this, there's no pointer? Is there a pointer? No, that's this thing here. All right, I'll just wave my hands. All right, so what are we developing? The technology is called Orb Cell, just our name for our stromal cell therapy. What's really important is that this is not a plastic adherent selected cell. This is a very homogeneous subpopulation of stromal cells that's positively selected, all right? So it's also allogeneic. It's like most in the field are beginning to recognize these cells. These cells don't have a regenerative capacity. They don't differentiate. We can't have them fill up with lipids and whatnot, but really not be differentiated at all. They're immunomodulatory. It seems to be preponderance of evidence in our hands and others work with these kinds of cells. They have immunomodulatory capability. So naturally, we're developing for indications that have an inflammatory immunomodulatory, an inflammatory component. The purification technology, or what it is that makes a difference, these are positively selected. So we found a marker on these cells that had not been previously characterized called CD362. It's also syndican-2. It's a well-known marker. It exists out there. The antibodies can be commercially obtained, but they hadn't been characterized in stromal cells. It turns out, I'll show you in just a few minutes, that all the modulatory activity in these stromal cell mixed populations of cells of, quote, MSCs that are plastic adherent, there's a population of cells that are 362 positive, and immunomodulation tracks with those cells. So we purify them. We just started earlier this summer. We now have 4 open clinical trials. 2 are about to start enrolling, the last 2, I should say, so all 4 are enrolling. All have been funded by grants from around Europe. We've raised about $60 million in European grants, UK grants, and a Wellcome Trust grant to develop these. There's been just an enormous amount of support amongst the great granting agencies for this approach to doing stromal cells. Make sure I get this. Okay, so, well, why, right? Well, so a lot of groups are moving just plastic adherent cells into the clinic, and we're not. The large reason is because both the FDA, member states of the EMA, not the EMA directly, as well as plenty of the scientific community, really concerned about the heterogeneity in plastic adherent stromal cell populations. And I'll tell you, MSCs aren't MSCs. It's a population of cells that stick to plastic that come out of bone marrow, adipose tissue, or cord. And there are a number of different cell types that are in there. That's well understood. In addition, there's also at least 9 different subpopulations of stromal cells in tissues, such as lymph tissue. There's maybe 5 or 9 in other tissues. So finding the actual population of cells that has the characteristics that we want is, we think, particularly important, and we've done that. So we get rid of all those extra, the pro-inflammatory cells, we get rid of the pro-adipocytes, the chondrocytes, fibroblasts, everything else that we know are in these populations, we end up with a very, very pure population of cells. That gives us a lot of lot-to-lot reproducibility, consistency. Also, having markers for the actual stromal subpopulations that we're interested in gives us an advantage in trying to understand mechanism of action, tracking the cells, and that sort of thing. And of course, we actually have several markers we've identified now that are, again, unique, proprietary, patent, all the rest. But so we actually have the ability to look at different populations of these cells. I will be honest, though, there's sort of an 80-20 rule of diminishing returns. You only want to over-purify cells to a certain point. We're getting cells to about 90 to 95% pure, and at the point, we don't have a good justification for trying to do a subpopulation of those cells. And of course, we have novel IP around it. So what actually is it? Well, like I said, it's an antibody-purified cell population we get directly from marrow, cord tissue, or adipose tissue. Our preferred tissue, like I think the last speaker, probably I'll show you in a second, but for obvious reasons, is umbilical cord. If you can get these cells from a tissue, then cord is a tissue to go with. We can select them by a number of ways, common, obvious ways. We tend to favor the owl Tito sorter. It's high throughput, fully GMP compatible, and benchtop. We use Terumo quantums for expanding the cells, but you can use Paul expansions. You can use plastic if you want. You can use microbeads and all sorts of things, but we tend to use the Tido and the Quantum. We've also used Clinimax for some of the early trials that we're involved right now. All of those processes are closable and semi-automatable. There's one step going from the Tido to the Terumo that we haven't closed because Terumo only makes a one-size-fits-all column, and we need a smaller column. We're working with them to fix that. When we fix that problem, the total system will be closed, and we can do this outside of a lower classification room, technically a non-GMP kind of space. It's just benchtop production. So it potentially lowers cost of goods even a little bit more. Anyway, so they're cryopreserved. They have very high viabilities and recoveries. It's like other MSC type products. We actually get a ton of product from one cord. In fact, we get about twice as many cells from a cord as we do from a marrow. It's not really a major point for today, but when you compare, it's hard to do. It's apples and oranges. If you compare a marrow process from a cord process, and if you notice we actually have 2 trials that are marrow-based and 2 that are cord-based at the moment. So we're just as motivated to make the marrow work and optimize it. That said, in the background of a huge field that works with marrow-derived cells, we get way more cells out of cord. They also grow better, they grow faster, and it's just a much more robust population of cells. So we're inclined to use that. The cells from either source, in about 7 out of about 11 animal models, work statistically better than plastic-adherent MSCs. Consistently, reproducibly, they just work better. In the other 4, you go, well, what happens in those 4? Well, in those, there's just not enough statistical power to show a statistical advantage. But in every single case, every study we've done with all our collaborators all over Europe, they always have a trend towards better. Not trend just means the bar looks higher, but this statistical error just doesn't really work out. You can't demonstrate it. But 7, absolutely, in all cases, they at least trend towards better. In 7 cases, they are definitely better. So it's a better cell, and we get more of them, and they grow better, and all the rest. So why umbilical cord? Well, it should be somewhat obvious. I think if you can get cells, they're discarded tissue. There's an infrastructure already established in the cord blood banking business to collect these under GLP, GMP conditions. There's quality systems, consenting process. We don't have to invest at all in that. They have a discarded tissue. They didn't know they had a product on their shelves, so we come in and say, we'd like to buy your cords from you, and they go, fantastic. And they just get them back out of the bin and wrap them up and sell them to us, sort of. But the end of the day is, we have a ready access to cords. Not only they are very inexpensive compared to marrow, but they're all age matched. So these are all 1 day old or 9 months old, however you look at it, versus marrow, where it's well accepted that robustness of the cells you get out of a marrow depends a lot on the age of the donor, and you can't get really young marrow donors. You really start in about mid-20s or so, which is pretty good. And you see a lot of irreproducibility or inconsistency from marrow donor to marrow donor. To be honest, we still see inconsistencies from cord to cord to cord, but we can bring in 10 cords to process 4 of them. We can easily go through them, screen them, take the best cords we want, or the best products out of those cords. And again, we get better yields and whatnot. So so that's nice. We can do it. Is there any good reason to do it? Well, this doesn't prove it, but this will show you why we like to select these cells out this way. If you consider that plastic adherence is a property of the cells that have immunomodulatory capability, it's also a property of other cells that do not. In fact, it's a property of pro-inflammatory cells and again all these other contaminants. But we can show you real briefly. I know there's a pointer here. Oh, that's not it. Right? How do I go back? Can I? Red button? That's what I just pushed. Oh, I see. Okay, we're fine. All right, so what we can show is that danger. Anyway, so what we can show is that the plastic adherent characteristics, colony-forming unit characteristic, tracks directly with CD362, not 271. CD271 is one of the canonical sort of markers, many groups will argue, is the marker for stromal cells. Well, it isn't expressed on cord-derived cells at all. They're negative. This is a marrow-derived sample, but you can see we can get towards the end on the right there. We actually get pretty close to 1-to-1 of plating. This was without any optimization of plating efficiency, Synthemax, or anything like that. So all of the plating efficiency tracks with the CD362 positive cells, not with the cells that are also that plate down but aren't 362 positive. So, fine. So this is a little out of order, but so we've got that. We can do this reproducibly. We get average around 90% if you look at all the preps we've done. We've done another several dozen since this slide's a little bit old. If you take out the outliers, lots we would argue didn't work. We're actually in about 95% pure of the cells that we would move forward with. So then does it really matter, right? So we can select, we can go through the motions, we can say we've done this, but is it really helping us? Well, in this regard, yes, in the sense that makes them pure. On the right is our selected cell. This is also from marrow. We're looking for 271 and 362. 362 is our marker that we're looking at. And you can see this just happens to be staining for pre-adipocytes. In the plastic adherent population, you can always find pre-adipocytes in there and other contaminants, only marking here for pre-adipocytes. If we take that plastic adherent population and take out our 362 cells, so this is what's left in a MSC population, you get an entire, just a ton of cells, and they're all contaminating cells. Right, so fine. So we know we're cleaning up these populations. We know we're getting them to near 100% purity. You still say, well, does it really matter? Right? Are we just jumping through some hoops for no good reason at all? So this is one of our 11 animal models, and it's a model that shows why we think this is important. We see this in several of the models, not all of the models, but we do see this several times reproducibly. This is one of our indications is diabetic foot ulcers, or neuroischemic diabetic foot ulcers that are non-healing. About 35 to 45% of patients, their wounds don't heal with good care. That's on the left. This is a rabbit model. It's an ear punch model in a diabetic rabbit. On the left is similar to humans. You get about 45% of the wounds do not heal spontaneously on their own. The leading therapy on the market is a product called Accelogen. It's a collagen matrix in collagen. You put that on, you get about an additional 20% closure. You get up to about 65%. Very similar to the human case. If you throw in plastic adherent cells into that, make a gamish of collagen and cells, you put it on, you get absolutely nothing at all. If you put on our 362 positive cells, you get another 20% increase, and now we're up to about 85% wound closure. We see a statistically relevant advantage. But interestingly, if you take that plastic adherent population of cells that did nothing, was neutral, take out our cells, again, you leave back those contaminating cells, addressing the question, do you need to, you actually see this. Again, in several animal models, you actually see a negative effect, right? So these cells, I won't say this is pro-inflammatory, but this is a clear negative effect of those contaminating cells in an MSC, standard plastic adherent population of cells. So when you see nothing happening with the plastic adherent cells and Accelogen, what's likely happening, because the 362 cells are in that population, you get a positive effect from the 362s, but it's buried by the negative effect of all the contaminating cells. So to me, that's kind of the justification for going with a purified population of cells. And this is what the FDA and member states in the EMA also think, is that there's not good things in standard MSC populations that are not being really well characterized. And this addresses that problem. So we have 4 open trials, as I indicated, listed here. I won't go through them all. Diabetic foot ulcers, diabetic kidney disease, autoimmune liver disease, also primary sclerosing cholangitis, and acute respiratory distress. And then we have another one with the Innovate UK grant that we picked up in about a year and a half. We should have a rheumatoid arthritis trial going. What's notable is, in addition, we got this 60 million or something in collective in money from all these grants. We got those because of the names, all the investigators. A lot of us here in the United States don't know these names, but these are the key opinion leaders in their respective fields. These aren't just good clinicians at a decent center. These are the folks that almost everybody in the field knows who they are. So these are really the big guns in their respective fields, which is how we managed to get these grants. But again, it underscores the consensus that this is the way to develop a stromal cell therapy. Everybody in the field wants to work with our cells. It's really kind of cool. And the regulators, we flew through the regulatory agencies in less than near record time to get through them for all the different trials. So the regulators like what we're doing, the PIs like what we're doing, the granting agencies like what we're doing. All we need to get is the investors to like what we're doing now. We're golden. And so the advantages overall, I'm just trying to rush this to get this to make sure we don't miss our buses. But we've really established a best in class. I mean, these are highly purified, homogeneous populations of a subpopulation of so-called MSCs that have immunomodulatory capability that are at least as good and trending better or better than plastic adherent cells in virtually every animal model we've tested these in, and that spans a spectrum of diseases beyond just the 4 that I showed you or the 5 I showed you here. We've demonstrated commercial scalability to do the trials. We have 4 academic GMP facilities doing our production, which is not our path forward, but it's because of the grants. These sites have different models for the different grants. We have upstream production at one, being distributed to downstream to other centers, then transferred to clinical sites. So we've made this as complicated and hair-raising as possible from an academic production perspective. And everybody, it's working beautifully. We've shown really nice comparability. The regulators loved what we were doing. And so we've shown transferability, we've shown scalability, and it's been, validate's not the right word, but it's been qualified and demonstrated feasible at least 4 different facilities around the EU. It's low source. We have a low-cost starting source, almost an unlimited supply of umbilical cords. There won't ever be a shortage of those, which much more consistency than we've even seen in our bone marrow products. So we have good source. We have better products, faster to expand. They expand faster. They expand more. Everything about it just speaks of it's the right way to go. And we have a lower cost of goods compared to marrow. And we also have begun to elucidate the mechanism of action of what these cells are doing, which isn't really commonly done. And we're just starting to elucidate. I'm not going to talk about that today. I can answer a question about it. Other groups now, we've published some of this. Other groups are now beginning to do similar work in their own unparalleled, and everybody's beginning to agree. The long story is it looks like this works through macrophages, polarizes macrophages, then elicits a Treg response. Which would be fantastic if that's the case, because it fits right into the whole immunomodulatory story. We haven't proven that yet, but that's the data that's coming together. We've got a nice body of data, but a lot more is to come on that. Again, this has been a very collaborative thing. All that grant money didn't come into our group. It came into consortia that's developing these cells with us. Groups all over Europe. Again, we're out of the University of Galway. That company is located in Galway, and we have a site now in Birmingham, the UK, where we're doing our manufacturing. So that's it. I want to thank you guys for listening. And I don't know if I went over. I'm right on the money, just about. So if there's any questions, I'd be glad to ask. Everybody wants to run and catch the bus. I understand. Any questions? Thank you.